All right, so we were talking last go around about Mendel and all of his work crossbreeding pea plants. And really quick, this is a recap of where we are. Mendel worked out, based on looking at the results of his pea plant crosses, over 19,000 over the course of several years, that there were some kind of things rattling around inside plant cells that determine what features the plants have. We now call those genes. Mendel just called them elements, but there's some kind of thingamabobs, thingamahoochers inside plant cells that ultimately determine how the plants grow and what they look like. Remember always, this is not just about peas. This applies to virtually all eukaryotes, including you. Genes may exist in several forms called alleles. Alleles may be dominant or recessive. In the examples I've shown you, every gene has two alleles. In the real world, genes may often have three or more, and we'll talk later about how that works. Each pea plant has two copies of each of its genes, and each plant got one gene from mommy plant and one gene from daddy plant. Those two copies might be the same, or each might be different. If a plant has two copies of a gene that are identical, we say that the plant is homozygous. And again, homo means same. Zygo means yoked together, linked in a pair. So two of the same thing linked in a pair is homozygous. If the two alleles of a gene are different, we say it's heterozygous. Alleles may be dominant or recessive, and the only thing that a dominant allele means is that it covers up the existence of a recessive allele. An individual that's heterozygous is going to have the dominant allele trait, and the recessive allele's effect will be hidden. And that's all that means. It does not mean common. Dominant doesn't mean better. It doesn't mean it's more common. It doesn't mean it's normal or anything like that. We can represent dominant and recessive alleles of the same gene by capital and lowercase forms of the same letter. So we can use capital P to represent the dominant allele for flower color, which is purple, and lowercase p to represent the recessive allele, white color. And we use P and P, big P and little p, not P and W, because we're trying to get across that these are two forms of the same gene. And so the alleles that any given plant has can be represented by a pair of letters. And last thing, when you work these out by hand, I often make a point of doing the dominant allele in block print, but writing the recessive allele in cursive, because otherwise it can be hard to tell, because a capital P and a lowercase p, other than size, look pretty much the same. And there's a number of letters where the lowercase is just a small version of the capital. And you want to avoid making a mistake because you couldn't tell what you just wrote. And we use this little calculating devices that were worked out by a guy named Reginald Punnett. They're called Punnett squares. And we can use that to predict the outcomes of crossbreeding experiments. So if we cross two pea plants that are both heterozygous, they have one dominant purple allele and one recessive white allele of the gene for flower color. We can represent that as capital P, lowercase p. Each parent passes on either its capital P or its lowercase p to each of its offspring, never both, never normally both. And so up here, we've got the this pea plant's alleles, it can pass on either capital P or it can pass on lowercase p, and we put those two possibilities at the tops of the columns. This pea plant over here can pass on either its dominant purple allele or its recessive white allele, never both. So we put those two options at the left of the rows, and then we fill in down and across. So here we move down that capital P from the top of the column and this capital P from the left of the row. And there we bring down the capital P from the top of the column and the lowercase p from the left of the row. And here we bring in the lowercase p from the top of the column and the capital P from the left of the row. And notice that the convention is that you write dominant before recessive. So we'd write capital P, little p, instead of little p, capital P. It's just, it tends to make things more clear. And then in this square, we bring down that lowercase p from the top of the column and the lowercase p from the left of the row. And so we put in this square little p, little p. You read this as meaning that 1/4 of all of your offspring, however many it is, 1 out of 4 offspring is going to be homozygous for the dominant allele. They'll be carrying 2 copies of the dominant purple allele, and they're going to look purple. 2 out of 4, which is of course 1/2 of all of your offspring, however many they are going to be heterozygous. They will look purple, but they will have 1 copy of the dominant purple allele and 1 copy of the recessive white allele. And then 1 of the 4 is going to inherit a recessive white allele, represented by lowercase p, from both parents, and that one will be homozygous recessive. 3 of these 4 lead to a purple trait, so 3/4 of your plants should have purple flowers, and that is in fact what Mendel found. 1/4 of the plants will have white flowers. Okay. This can come in handy when you start looking at crosses where we have to deal with 2 pairs of genes. Up here at the top, that is a smooth yellow pea, dried. They all look wrinkled, I think, when you boil them a bit, but when they're dried out, some peas have, some plants produce smooth peas and some plants produce wrinkled peas. And I kind of intensified the colors a little bit. They're not quite this bright, but some plants produce yellow peas and some plants produce green peas. So what happens if you take a plant with smooth yellow peas and you crossbreed it to a plant with green wrinkled peas? Mendel had already worked out that smooth is dominant to wrinkled. There's a gene for pea shape, and the smooth allele is dominant to the wrinkled allele. And he'd also worked out that yellow pea color was dominant to green pea color. There's a gene for pea color, and the yellow allele is dominant to the green allele. He'd already shown that. And incidentally, if I were to give you a problem like this, I would give you that background information. So the genotypes we're going to cross look like this. The yellow smooth parent is homozygous for the dominant yellow allele for pea color. So we write that as capital Y, capital Y. And it's homozygous dominant for the smooth pea shape allele. And we write that as capital S, capital S. The other parent is homozygous recessive for both green pea color and wrinkled pea shape. And we write that as little y, little y, little s, little s. I've made the little s italic because otherwise it would look like a smaller version of the big S. But I do want to make sure that you see they're truly different alleles. Each parent can contribute one of each pair of alleles that it's got, not both. And in this case, this is not very interesting. Each pea plant can only pass on one of each pair of its alleles. The yellow smooth parent can only pass on one dominant yellow allele and one dominant smooth allele, which we write as big Y, big S. And that's the only option that the yellow smooth parent can pass on. The green and wrinkle parent can only contribute the little y and the little s alleles. So every egg or sperm of the smooth yellow pea is going to be carrying the allele combination big Y, big S. And every egg or sperm produced by the wrinkled green parent can only carry the allele combination little y, little s. And this sets up a very boring Punnett square in which all of the F1 offspring got one dominant yellow allele and one dominant smooth allele from the yellow smooth parent. And they got one recessive green allele and one recessive wrinkled allele from the other parent. All of those offspring have the genotype big Y, big S, little s, and they all have the phenotype smooth yellow peas. Remember, genotype is the description of what alleles an organism has. Phenotype is a description of what it looks like. So if I ask you an exam question, what is the genotype? The answer is going to be some combination of letters. If I ask you what is the phenotype, the answer is going to be smooth wrinkled peas or tabby stripes or sickle cell anemia or leaves with a white V shape in them, something like that. Phenotype always refers to an observable trait. Genotype refers to the alleles that an organism is packing. This gets a little more interesting if we were to take two of these F1s. We will ignore the fact that we'd basically be mating brother and sister pea plants. If you do that too much, your peas are going to be playing banjos. But if we cross two of the F1s with each other, it gets a little more interesting because each of these F1s is heterozygous for both genes. Each one of these F1s is carrying a dominant yellow and a recessive green, and a dominant smooth and a recessive wrinkled allele. And that makes things more interesting because each parent can contribute one of each pair of alleles that it has. And there are 4 possible allele combinations that each parent can pass on. I'm going to show you something that you might remember from algebra class. If we have this genotype over here that's big Y, little y, big S, little s, well, we have 2 pairs of alleles, right? 2 Ys and 2 S's. A plant that's got this combination of alleles, any given egg or sperm that it makes might pass on the first of each pair, or the outers of each pair, or the inners of each pair, or the last of each pair. Does anybody remember FOIL from 8th grade algebra, right? Remember, that's how you learn to, multiply binomials, right? If you're multiplying x plus 5 times x plus 2, you multiply the 2 x's, then you multiply the outers, x times 2, 2x, and then you multiply the inners, 5x, and then you multiply the lasts, and you get x squared plus 7x plus 10. Anybody remember that FOIL? It works just as well here. If you have this genotype, each parent can pass on one of the Ys and one of the S's, and it can pass on the first of each of these pairs, or the outers, or the inners, or the lasts. This is a quick way that you already know to work out what allele combinations each parent can pass on. And that sets up a 4x4 Punnett square that's a little bit more complicated, and it looks like this. And we fill it out by taking those letters and just moving them from the tops of the columns down, and from the left of the columns to the right. And notice that when we fill these out, we sort. So we write big, like in here, this says big Y, big S, not big Y, big S, big Y, big S. It's much easier to interpret if you sort the letters as you're filling in the blanks of the Punnett square. You've got a sheet of practice problems already, and there's more coming. So with a little practice, you should ultimately get the hang of it. Now, how do we read this Punnett square? Well, those 9 right there, hey, I can make it flip back and forth. Look, it's computer animation. Okay, those 9 have at least one dominant allele for both genes. Each one of these has at least one capital Y and at least one capital S. So they're going to look yellow and smooth. That's their phenotype. And they, all 9 of them, since they've got one copy of each dominant allele, they're going to look yellow and smooth. 3 of these have at least one dominant Y allele, but they are homozygous recessive for the wrinkled allele. And that's this one, and that one. And they're going to look wrinkled, and they're going to look yellow because they've got at least one dominant yellow allele, but they are homozygous recessive for the wrinkled allele. So they're going to look yellow and wrinkled. And yes, that is what a yellow and wrinkled P looks like, although I did Photoshop boost the color intensity a little bit to make it obvious. Okay, that leaves 4. These 3 have 2 copies of the recessive green allele, which we write as little y, but they've got at least one copy of the dominant smooth allele. So that one, and that one are going to look green but smooth. Ta-da. And that leaves 1 out of the 16 that is going to be both wrinkled and green. And it will look like that. So 9 out of 16 offspring, that works out to 56.25%, right? Just a little more than a half of those offspring are going to look yellow and smooth. They are going to have at least one copy of both dominant alleles. 3 out of the 16 are going to look yellow and wrinkled. They're going to have at least one copy of the dominant yellow allele, but they will be homozygous recessive for wrinkling. 3 out of 16 are going to look green and smooth. They will have at least one copy of the dominant smooth allele, but they will be homozygous recessive for the green allele. And only 1 out of 16 will be both wrinkled and green. So there, that's how you can do this with looking at inheritance patterns for 2 different genes at the same time. You can increase that. You could do an 8x8 Punnett square to look at the results of crossing organisms where you're looking at 3 different genes at once, or a 4x4 Punnett square to look at no, that would be a 16x16 Punnett square to look at the effect of 4 genes at once, and so on. Although looking at large numbers of genes like that, we've actually got more convenient ways of doing the calculation. but the Punnett square will work for what we need to do in this course. All right, checking my time. Okay, so Mendel announced his findings in 1865, and he published them in 1866. He published them in a scientific journal. Remember that when scientists make discoveries, they usually don't go straight to newspapers. They usually don't write books. They publish articles in rather specialized magazines, which used to be all print, although these days almost all of them are either electronic only or printed in both ways. These are called journals, and he published his findings in a journal called the Proceedings of the Natural History Society of Brno. In other words, it's not exactly Time magazine. It was kind of an obscure place to publish. He did send copies of his work to other people, but they didn't really understand what he'd done. And his work was mostly forgotten for the next 35 years. He did try to keep going, trying to study inheritance in other species of plant to try to test whether his discoveries were really universal. Unfortunately, he got kicked upstairs. He was elected abbot of the monastery in 1868. He had to spend the rest of his life administering the monastery's affairs and getting into political tensions with the government of the Austro-Hungarian Empire, which I do not fully understand what he was up to, but he never did any more science, unfortunately. And he died in 1884. A Dutch chap by the name of Hugo de Vries, independently of Mendel, several years later, started doing some breeding experiments with a different flower, the evening primrose, a rather attractive European garden flower. And it just so happened that he rediscovered some of Mendel's findings. Like Mendel, he argued that traits must be inherited as units rather than as blends. Heredity was due to some sort of elements or particles rattling around inside cells, not like mixing paint or other fluids. Those units that he hypothesized must exist, he referred to as intracellular pangenes, which we have just whittled down to genes. So he's the guy that coined the word. And in 1900, de Vries and a couple of other guys named Karl Karens and Hugo Czermak, independently of each other, happened to find Mendel's original paper, which had been gathering dust on library shelves all this time. And they understood what it was that he'd done. And understand what he did, it doesn't seem like very fancy science. I mean, his main experimental apparatus was a garden and a bunch of pea seeds and bits of sealing wax and a paintbrush and a razor blade. You know, it doesn't look like he did that much, and it looks like he basically just counted a whole bunch of bleeping pea plants. Not the most exciting thing you'd ever see. And yet, he's worked out something critically important about how heredity worked. And this is a beautiful case of rattling that sealed cardboard box and deducing what's inside. And de Vries and other scientists went on to do experiments with other plants. They looked at animal breeding, they looked at human breeding, and revealed that Mendel's basic laws apply to virtually all eukaryotes, including you. The same rules that govern whether two pea plants are going to produce purple or white flowered offspring govern the rules of whether a man and a woman who are having a baby are going to have a baby with muscular dystrophy or multiple sclerosis or Tay-Sachs syndrome or sickle cell anemia. Right? This applies all over the place. There are a few new wrinkles, and I'm going to introduce a few of those in the time that we've got. We're a little bit behind where I meant to be, but it's probably just as well. One of the first things that was discovered when Mendel's work was rediscovered is that alleles do not necessarily come only in two forms. A lot of the examples that I'm going to give you in the rest of today's lecture and more on Monday are going to revolve around kitty cats. That's not so much because I love kitty cats, although I do really like kitty cats, preferably medium rare. Sorry, one of them peed on my things last night. I'm not in a good, we're not friends right now. Cats. Anyway, but cats are something you've seen before, and cats are something that you might feel a little bit more of a connection to than pea plants. And a nice example of alleles coming in more than two forms is in cats. There is a gene that governs how much dark base color the cat has. Now, the base colors of a cat are relatively few. You can have black, dark brown, light brown, or orange, or various combinations of those. And there's other effects that are possible that we'll get into. But it turns out there's one gene for the presence of a dark base color, and it comes in three alleles that we can abbreviate capital B, lowercase b, and lowercase b prime, B with a tick mark next to it. That prime mark looks like a, looks like a single quotation mark. And the capital B stands for black, and black is dominant to medium brown, which we represent with lowercase b, and light brown, which we represent by little b prime. Black is dominant to medium brown and light brown, and medium brown is dominant only to light brown. Now, since these are cats, I should refer to these as medium brown and light brown. But cat breeders refer to these colors as chocolate and cinnamon. And incidentally, what we call black, if you've ever seen a black cat in really bright sunlight, it turns out that a black cat is really, really, really dark brown. We have a couple of black cats, and when you see them lying in a sunbeam, you can see the brown highlights on their fur. But it's dark enough that we usually call it black. So up at the top left, that's a black cat, not one of mine. And at the lower left, that is a dark brown or chocolate cat. And at the lower right, that is a light brown or cinnamon cat. Now, this black cat could be homozygous for the dominant black allele, or heterozygous and have one black allele and one dark brown, or be heterozygous and have one black and one light brown allele. It doesn't matter if what the cat is heterozygous for. If it's got one black allele, that will cover up the presence of any recessive allele for fur color. So we can't tell this cat's genotype just by looking at it. All we know is that this cat has one copy of the black allele at least. The cat down there could either be homozygous for the dark brown allele, or could be heterozygous with one copy of the dark brown and one copy of the light brown. So we can't tell that dark brown cat's genotype. But this cat right here with the cinnamon fur can only have the genotype little b prime, little b prime. The only way you'll get a cat with cinnamon brown fur with light brown is for that cat to be homozygous for the recessive light brown allele. And many, many, many genes have more than two alleles. This happens all the time. Each cat can only have two of the two alleles, right? So you can't have a cat that's big B, little b prime. because cats only have 2 of each allele, but there can be more possible alleles that each cat could have 2 of, if that makes sense. I hope it does. An example that you're going to work with in lab coming up fairly soon is the ABO blood group. This was discovered in 1900 by a guy named Karl Landsteiner, who won one of the first Nobel Prizes in medicine because his discovery made blood transfusion possible. There are 3 alleles that govern your ABO blood group. You've actually got about a dozen different blood groups, but the one that's most important for ensuring safe blood transfusions is the ABO blood group. And in this, your blood type might be A or B or AB or O. I have type O blood. Some of you may have any of the other 3 types. And there's a wrinkle here in that the A and the B alleles are codominant to the recessive O allele. A is dominant to O, B is dominant to O. And if you're heterozygous with one A and one B, you're essentially both at once and you have blood type AB. So if you have type A blood, you might have the genotype AA, or you might have the genotype AO. If you are blood type B, you could have the genotype BB, or you could have the genotype BO. Ew, BO. If you have type AB blood, your genotype is AB, one copy of the A allele and one copy of the B allele. And if you have type O blood, your genotype must be OO. You'll work with this a lot more in an upcoming lab. I will only point out that this has been used not just in assuring safe transfusions, it's been used in things like paternity testing. You can't prove that so-and-so was the father of a certain child, but you can in some cases prove that so-and-so was not. And you can sometimes rule that out using Mendel's laws and the inheritance of blood types. And again, you're going to see this in more detail in an upcoming lab. There's another wrinkle called incomplete dominance. And for the moment, I'll step away from cats and show you some garden flowers known as snapdragons. And in this case, if you cross a red snapdragon with a white snapdragon, the offspring all come out pink. That's not what we saw when looking at Mendel's peas. Mendel crossed purple and white peas and got all purple offspring, nothing in between purple and white. But here we do get something that's in between red and white. It's pink. And it turns out that if you cross 2 pink snapdragons, you get, in the offspring that you get, you'll get 1 red and 1 white for every 2 pinks. 25% of the offspring are going to be red, 50% will be pink, and 25% will be white. And the reason is that in this case, the red allele is incompletely dominant to the white allele. The red allele does not completely cover the presence of the white allele. You could think of this as some of the white color peeking through, if you like. So heterozygotes turn out to be pink. This is still following Mendel's laws. The difference is that a red snapdragon is homozygous for the red allele. The pink snapdragons are heterozygous and they look pink. They don't look completely red because red is incompletely dominant. And then the homozygous recessive snapdragons just look white. So there are cases where a dominant allele does not completely mask a recessive allele. And we call that incomplete dominance. In kitty cats, there is an allele for white spotting, and it's incompletely dominant to the allele for solid color. And we can write this as capital S for white spotting and lowercase s for solid color. All three of these cats are basic black cats. They've got at least one copy of that dominant black fur allele, but their color is also affected by the alleles that they've got at the gene for white spotting. A cat that has two dominant alleles of the white spotting allele, or of the spotting gene, like the one seen on the left, that cat is so covered in white spots that it's a mostly white cat with just a little bit of black. The cat on the right has two copies of the recessive solid color allele, and this cat is solid all over. The cat in the middle is a heterozygote, and it's in between the two homozygotes. It has mostly black with just a white spot on its face. We've got one of our cats who was snuggling up to me before the start of class, but has since vanished. One of our cats is almost completely black except for white socks and white on his face. They call this a tuxedo cat. Another one of our cats is almost completely black except for one tiny white spot on his belly. Some cats might be completely black except for one little white star on the chest or something like that. So this is incomplete dominance. A cat that is homozygous for the white spotted allele has so many white spots that it's mostly a white cat. A cat that's homozygous recessive for the solid color allele is going to have no spots at all. And the in-between cat is heterozygous, and it will show white spots, but just a few, maybe just white socks or a white star or the tuxedo pattern. Okay, the last wrinkle I want to hit you with is called epistasis, and that is what we call it when several genes affect the same trait. You're looking at a black cat. So that cat has maybe genotype big B, big B. The basic color is black. This cat also has two copies of an allele for dilute fur color. And it is recessive to the non-dilute allele or dense allele. And if a cat has two copies of that dilute allele, then whatever the cat's base color is, black, brown, it doesn't matter, whatever the base color is, that color is going to be lightened a notch, and you're going to end up in this case with a gray cat. There's no gene for gray fur in a cat. This cat is showing black fur, but that black fur is modified by alleles of a different gene that cause whatever the base color is to be diluted. And we call that epistasis, several genes affecting the same trait. So on the left, you've got a black cat. On the right, you've got a chocolate brown cat, a medium brown cat, but both of them also have the homozygous recessive genotype, two copies of the allele for dilute color, and that takes their base coat and lightens it several notch, producing a gray coat color and a tan coat color, which cat breeders for some reason call blue and lilac. If you've ever seen blue cats, a good friend of mine has some beautiful Russian blue cats. And what they are is black cats, but they've also got two copies of this dilute allele. So they're black cats with the color lightened a notch. So the effect is that they look kind of a steel gray. And that happens a lot. There is lots of epistasis in this world. Last I looked, there were at least 9 different human genes that had a noticeable effect on your skin tone in humans. Each of those genes has at least 2 or more alleles. All of those genes contribute something to human skin color. And if anybody was curious, we talk about people as being black or white, ebony and ivory and all that. The truth is that we're a range. We go from everywhere from, pasty flesh goth, all the way to, deep mahogany. And most of us are on a continuum in between those extremes. We don't really come in nice, neat skin color classes the way that pea plants come in either purple or white. And the reason is that there's a huge number of possible human genetic genotypes just for the genes affecting skin color. And these genes interact with each other and affect each other in very complicated ways. Skin color is very highly epistatic. We have lots of genes that cover it. You can't really do Punnett square. Well, you could, but you'd need for 9 genes, you'd need, I think, a Punnett square that was, I think, hang on. I think you need a 1024 by 1024 Punnett square to look at the inheritance of 9 different genes all at once. So we can't really use Punnett squares to track inheritance like that. And that makes human skin tone a continuum. We don't fit into nice, neat classes the way that pea flowers can be classified as purple or white. And this is true for an awful lot of human traits. Your build, your height, your overall risk of getting cancer. All of these have some degree of genetic influence. They're all affected by the environment as well. Your skin color is affected by your genes, but it's also affected by how much sunlight you've been getting or how much time you've spent in the tanning bed. And they're affected by so many genes that it's very hard to use Mendelian methods to track their inheritance. We can't always predict what we're going to get. I was reading recently that they've isolated at least 200 human genes that have some effect on athletic performance. So that's really complex. Athletic performance does tend to run in families, like, Archie Manning and his kids, for example, or LeBron James and his kid. But there is no single gene that determines whether you're going to be an Olympic gold athlete or a couch potato. And you need to remember this because we keep looking for the gene for such and such a condition or trait. Oftentimes there's a bunch of genes interacting with each other in complicated ways. So the real world does not always fit Mendel's nice, neat, and tidy little mold, which is okay. Mendel's work was an important beginning, an important foundation. In many cases, we've been able to go beyond it. And that's the way science goes. And I'm over time by 1 minute, so I'm going to go ahead and stop the recording right now.